chain, or ETC. Okay? And so, like I said, we're going to talk about what's happened up until now. And so we know that in the process of cellular respiration, we had to start with glucose first. So we're going to start up here with glucose at the top. Glucose is C6H12O6. Okay? Glucose went into the process of glycolysis. And that process of glycolysis produced 2 NADH, and it also produced 2 ATP. Right? One of the other things that it produced was a molecule that's called pyruvate, and specifically it produced 2 of them. Okay? Those pyruvates then go on to be processed further, and they get processed further in a process called the preparatory steps, or the prep steps. And the prep steps are going to form some compounds. They are going to form 2 high-energy electron carriers in the form of 2 NADH. No ATP formed in the prep steps, but they are going to form 2 molecules called acetyl coenzyme A. So we're going to form 2 acetyl CoAs. Then we're going to go from the prep steps into the Krebs cycle. The Krebs cycle will produce 6 NADHs, 2 FADH2s, and then also 2 ATP, which actually is GTP. We'll put it there. They're GTPs. But for all intents and purposes, we just call it ATP. Okay? So, that's where we are up until now. Everybody agree? This is all the stuff that we've produced. Total of 4 ATP-like things, and 10 NADHs, and 2 FADH2s, right? Yes? So total here, when we go this way, these are all going to go into the electron transport chain. And those are going to become the inputs to the electron transport chain. Right? Yes? All right. Let's talk about the ETC then. Electron transport chain. First thing we need to know about the electron transport chain is we need to know where it's happening. So we're going to draw over here a little mitochondria. It's the outer membrane of the mitochondria. The inner membrane is this folded up thing, right? Okay. We said that glycolysis was a process that happens outside of the mitochondria. The prep steps and the Krebs cycle happened in the mitochondrial matrix, which is the area on the inside. And then embedded in the membrane is where the ETC goes on. Okay, embedded in that inner membrane. Now the H+ ion gradient is going to build up in the space in between the inner membrane and the outer membrane called the intermembrane space. And all of these reactions that we're pretty much going to talk about are happening in the matrix, which is the area on the inside. Okay? All right. So let's draw a picture then of the inner membrane. Here's phospholipid bilayer. And then we're going to have these things, these protein complexes. And all the protein complexes have funky names. And so it's not really necessary for us to know the names of them. What was that noise? Is that a phone? Yeah. That sounded like actual like metal clanking around or something. All right. So they have funky names, so we don't need to know the names of them. We're just going to refer to them as protein complexes, and then we're going to give them numbers. So we're going to say they're protein complex 1, 2, 3, and 4. Later in the year, we're going to talk about this protein called a cytochrome that's used for determining like ancestry and stuff like that. And it's one of the proteins that is involved here. Cytochrome C is going to be found in protein complex 3 and in protein complex 4. Okay, so we're going to refer to these as protein complex 1, protein complex 2, protein complex 3, and protein complex, Roman numerals are hard, 4. All right. At the end of this, then, there's going to be one more protein, and this protein we do know its name. This protein is one that we've talked about a lot. We usually draw it looking like an Erlenmeyer flask here, and it is ATP synthase. And we know ATP synthase has channel running through it, and that channel is going to allow H+ ions to flow through, and then we'll create ATP. Now, to my knowledge, there is no exact efficiency here known for ATP synthase, but we are going to use the efficiency of about 3.3 H+ ions per ATP produced. Okay, roughly that amount, 3.3 H+ ions per ATP produced. All right. So this is, again, this is ATP synthase. Okay? This is embedded in the inner membrane of the mitochondria. So basically what happens here is we are going to take the electrons, the high energy electrons from our two high energy electron carriers, which are NADH and FADH2, and we are going to enter them into this chain of protein complexes. And different protein complexes are going to do different things. Okay? So the first thing that we need to know is that we are going to dump the electrons from NADH into this first protein complex, and that's going to produce what molecule then? If we NAD+. Yep. So now we've got two high energy electrons in here. These high energy electrons, when they enter, are going to be used to pump 4 H+ ions. Okay, so we'll do the H+ ions in green. 4 H+ ions are going to get pumped into this space here, which I guess we should have labeled. This is the intermembrane space. And this is the matrix, the mitochondrial matrix in here. And that makes this the inner membrane, right? So we're pumping 4 H+ ions. When we do that, we are going to use up some of the energy in our electrons. So we're going to draw an arrow going down to represent the fact that our electrons have lowered in their energy level, right? Now, we'll talk about a little bit later why this second protein complex is important here. But for now, it's not really important, okay? We're going to say that at this point, the electrons are passed along here to the next complex. Okay? Now, when they go into this next complex, their energy is used to pump 4 more H+ ions. Okay? So we've pumped a total of 8 H+ ions then with NADH, using the 2 electrons per NADH. Following so far?
How much NADH?
We just 1 NADH. Because each NADH has 2 electrons, and so we've used 2 electrons total, which is just 1 NADH.
And it's the same 2 electrons, just maybe. Okay.
Just moving along the line. Okay? Now, when they get to the end, they are low energy electrons, right? Now they are sad, low energy electrons. Okay? So, there's going to be a traffic jam here of electrons where all these low energy electrons are going to build up in this complex, in complex number 4. And that's bad because then that won't allow any more electrons to travel down the electron transport chain. So there has to be a place for them to go. Okay? And the place for them to go has to be something that is willing to accept low energy electrons and is able to take electrons away very well. So we're going to need something with really high electronegativity, like oxygen. This is why we say that oxygen is the terminal electron acceptor. So we're going to need one molecule of oxygen here. Okay, one atom of oxygen. But atoms of oxygen don't really exist by themselves. So we're going to have O2, which is a molecule. We're only going to need one oxygen. And so the way that we write that is kind of tricky. It's 1/2 O2. Okay, meaning we're just going to need one part of the O2 molecule. One oxygen total. That's going to combine with the 2 electrons. It's also going to combine with 2 H+ ions. And it's going to form then what? 1/2 O2 plus 2 H pluses is H2O. Now, this in itself does not pump H+ ions, but it does contribute to the H+ ion gradient, right? And you can see that because even though you're not pumping H+ ions here, this is removing H+ ions from that space, right? You're taking H+ ions away from the mitochondrial matrix. Yes?
There's an abundance of H+ ions in the mitochondrial space?
Yes, there's H+ ions everywhere. You should just always assume that there's H+ ions present. That's because of the self-ionization of water. Anytime that the pH goes below a certain amount, above a certain amount, they're going to ionize into hydrogen ions. And
where are they taking
hydroxides.
the H+ ions from?
From the matrix, which is going to lower the concentration of H+ ions in the matrix, which then raises the gradient, right? All right. So that means that this can be considered moving 2 more H+ ions, right? Even though it's not technically moving them. What's up?
What happens to the other oxygen?
The next NADH uses it.
Okay, so it just kind of sits there for a little while?
Really, what's more correct is that 4 electrons can end up in your final thing here, and then together those 4 electrons can take in 2 oxygens, right? So nothing happens until there's 4 electrons available, and once there's 4 electrons available, then one of these reactions happens. Okay? So that means each NADH pumps how many H+ ions? 10, right? 4 here, 4 here, 2 there, right? So per 1, we can say that we have a ratio here of 1 NADH is able to pump 10 H+ ions. Okay? But the difference is FADH is going to enter at a slightly different spot. Because we said that the electrons on FADH2 were slightly lower energy, that means that it's going to have to come in at a lower energy spot. And we said that this second thing really didn't do anything. That's its job, is to equalize the energy of the electrons in NADH once they've moved their first H+ ions, and when FADH2 comes in. So FADH2 enters here and then turns into FAD when it dumps its 2 high energy electrons in. Okay? So if FADH2 enters its electrons right here, it skips this first ability to move H+ ions, so it only pumps 4 here and then 2 at the end. So how many does it do? 6. So we can say that 1 FADH2 pumps 6 H+ ions. Yeah.
When you say pumps, where is it pumping?
They're going into the intramembrane space.
Okay.
Yep.
But how can it pump 6 if the NADH already pumped 10? I mean,
I don't know what you mean.
because I don't know.
There's always H plus ions in there. Think of like infinite H plus ions. Whenever you need them, they're there. Okay? Okay. So, we can do some maths now. All right? And we can say, okay, we're going to use all the things that we know. So we started with 10 NADH, right? That was from this part. We said that in total, we would have 2 plus 2 plus 6, which is 10. So 10 NADH enter. We said that every 1 NADH pumps 10 H plus ions. We said that every 1 H plus ion, or sorry, no, not 1, 3.3 H plus ions will produce 1 ATP as they flow through ATP synthase. Okay. And then for FADH2, we said that we had a total of 2 FADH2. We said that every 1 FADH2 produced, or was able to pump 6 H plus ions. And we said that it was 3.3 H plus ions. And that's really just an approximation per 1 ATP. Okay, so then we can go through and say, okay, get out my trusty calculator. And we say 10 times 10 divided by 3.3. We'll call it 30.3. Oops. ATP. Okay. Then we'll say 2 times 6 divided by 3.3. And that's, we'll call it 3.7 to make our life easier, even though that's not going to be exact. You can't like point something ATP. So we'll say that's 3.7 ATP. Okay. So together, how many ATP are produced there? 34 ATP. So now we can look at our inputs and outputs. So we said we had 10 NADH, right? We had 2 FADH2. Okay, does anything else need to be input? H plus ions, we don't need to consider those. Oxygen. Oxygen needs to be input here. Okay, so the question becomes, how many oxygen atoms are required per molecule of NADH or FADH2? What? 1/2 O2, which is what? 1, right? So a single oxygen. So how many total things do we have here? 13? Oh, no. Okay. So if it's 1 O2 per molecule here, and how many molecules of NADH and FADH2 do we have? 12. So that's going to be 1 atom of oxygen per one of these. So that's going to be 6 O2, right? Which is 12 oxygens, right? Did I lose anybody on that? Yeah, each of these, each NADH requires 1 oxygen to take the electrons at the end. Each FADH2 requires 1 oxygen, right? So that means that if we have 12 total molecules here, we're going to require 12 total oxygens, and that's 6O2, right? Okay. The other thing that we probably need to include here, because we included it in the rest of the stuff, is we're going to need 34 of what? ADPs. Okay? So our outputs then are going to be 10 NAD plus 2 FADs are 6O2. How many waters does that make? Think about this for a second. Before you say anything, just it's O2. How many oxygens per water? 1. So 12. Okay? So then our 34 ADPs become 34 ATPs. Okay. So, because we did it on the rest of them, we'll do it on this too. The NADs go where? We're just going to say they get recycled because they're going to be used in all the other parts. They're going to be used in glycolysis, they're going to be used in prep steps, and they're going to be used in Krebs. Okay? Where do the FADs go? Yeah, they're Krebs only. So recycled, we could say recycled into the Krebs. Krebs. Okay? H2O. So it's waste plus recycled. We'll talk about that in a second. And then ATP can be used for what? Anything. Cellular processes. All right, so let's make this all work. How much time do we have left? All right, we're gonna do this. We're gonna do this the super good way. What's up?
So the picture that we drew of the actual electron transport chain, that's on the little diagram where they all occur. That's on the that's embedded on the outside.
It's embedded in the inner membrane. Okay, here we go. We're gonna do this the super big way. Okay, so here we go. We're gonna do reactants on this side and products over here. And I'm gonna put a big line in. So let's start with NAD plus, because we said that was going to be a reactant, right? Let's go through here. Just in our inputs. We're just looking in the input spot now. Okay? How many NAD for glycolysis? 2. How many NAD for prep steps?
2.
How many NAD for Krebs cycle? So that's a total of how many? 10. Okay? So times 10. Okay? Was any NAD plus produced in anything else other than the electron transport chain? No, it was never an output other than in the electron transport chain. How many did we make? 10. Okay? Right? Our output there of 10. Right? Let's go with FAD. How many did we use? FAD was only in the Krebs cycle, right? So that was only right here. We used 2. How many did we produce in the electron transport chain? 2. Okay? Next, let's talk about ATP. Any reactants of ATP? Well, any ATP in the reactants?
Glycolysis.
Glycolysis. Okay. So that means that we're going to have 2 there. Okay, let's talk about how much ATP was made. So glycolysis makes how many? 4, right? Any made in prep steps? No. Any made in here in the Krebs cycle? No. Let's say that GTP is ATP. Yeah, so 2. And then how many do we make in electron transport chain? 34. So that's a total of 40, right? Okay. Let's talk about ADP. Actually, let's not worry about ADP. All right, we'll do ADP, but it's gonna get messed. Nah, it'll be fine. Okay, it'll be fine. ADP, yeah, it'll work. I have faith. All right, how many ADPs in glycolysis? How many in the outputs? Okay, so keep that in mind. So 4 in the inputs. How many ADPs in prep steps? 0. How many in Krebs?
Or are we gonna count GDP as
We're gonna count. Yeah, so we're gonna count GDP. So total of 6 here. Okay, and then how many ADPs here? 34. So that's a total of 40. Okay, and then ADPs here. The only time that we produced ADP was in glycolysis, and we made how many? 2. Okay. What's that? It was clean. Yeah, it'll work. All right. So let's look at oxygen now. There's a lot. Okay. Where do we use oxygen? Electron transport chain. How many? 6. Okay. Do we produce any oxygen? No. Okay. What about water? Where do we have water as a reactant? Krebs cycle. That was how many waters in Krebs?
4.
How many waters in prep steps?
2.
2. So that's a total of 6. So we'll have 6 waters. How many waters do we produce? Here, 12, right? All right, how about C6H12O6? Okay, one of those. And then how many, oh, I didn't get down enough. Boom. How many CO2s did we produce? Where do we produce CO2s? 4 here in the Krebs and 2 in the prep steps. So that's 6. CO2 times 6. All right, ready? So we will cancel out 10 NADs with 10 NADs. 2 FADs with 2 FADs. We can't cancel out anything here, right? We can cancel out, I guess, 2 of the ADPs to make 38. We can cancel out 2 of the ATPs to make 38 ATPs. Right? O2 doesn't cancel. We can cancel out 6 of the H2Os to make 6 H2Os over here. We can't cancel out the C6H12O6. We can cancel out the CO2, or we can't cancel out the CO2s. So our equation becomes C6H12O6 plus 6 O2 plus 38 ADP yields 6 CO2 plus 6 H2O plus 38 ATP. That's it, right? That's the thing that we know. C6H12O6 plus O2 yields 6 O2 yields 6 CO2 plus 6 H2O, and we make 38 ATP in the process. The difference here is the only thing we didn't talk about is to get the pyruvate into the mitochondrial matrix. It takes 2 ATP to do that, and so that's why we have 36 ATP in eukaryotes, but 38 ATP in prokaryotes. Make sense? Did we do it?